Okay, so welcome everyone, and thank you for virtually joining us for the latest version of the Variant Effect seminar series. My name is Diego, and I'm a member of the VES organizing committee, along with Stephen Airwood, Medea Soma, Jan Obudu, and Adrini D'Souza. These seminars are made possible with support and a lot of help from Laura Muffley and Alex Hopkins, among several others, and our sponsor, the Atlas of Variant Effects Alliance. So a big thank you to them. As a reminder, we are always on the lookout for potential speakers for future seminars. If there's anyone you'd like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, just before starting the presentations, I have some housekeeping items to mention. Each talk is about 20 to 25 minutes, so there should be some time for questions. Please post those questions at any time to the Zoom Q&A, and we'll read them out to the speaker at the end. If you have additional questions after the end of the seminar and you're an AVE member, feel free to visit our seminar series Slack channel for more discussion. And finally, we're posting updates on our Twitter account, so be sure to follow us there at Variant Effects. You can catch the seminar today and previous presentations that have been recorded on our affiliate CMAP YouTube channel. Don't worry about trying to copy these URLs. I'm going to post links to the chat once we get started. All the details of this seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. And for the last housekeeping item, this is rarely an issue, but just as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. So without further ado, we have two fantastic speakers scheduled for today's presentation. First up, Dr. Sofia Battaglia is a postdoctoral researcher in Brad Bernstein's group at the Dana-Farber Cancer Institute and the Broad Institute. Her work focuses on developing and applying novel technologies for understanding long-range interactions of regulatory elements of complex loci. For this work, she uses ultra-long-read epigenomic profiling of single DNA molecules. The title of Sofia's talk today is Single Molecule Regulatory Landscapes. Sophia, feel free to start sharing your screen and go ahead and start whenever you're ready.
Okay, now I am unmute. Share screen. So can you see my screen now?
Yep, looks good.
So, okay. So hi, everyone. I'd like to thank again the organizers for the opportunity to speak here today. My talk title is Single Molecule Regulatory Landscapes. And I'm going to share some very recent work where we use nanopore sequencing to profile this ultra long DNA molecules. So it's no news to this audience that variants can affect the activity of regulatory elements and gene expression, and that the effect of variants is cell type specific. And so to better understand the variant effect, we also have to know which enhancers regulate which genes in which cell types and cell states. And as we know, gene regulation is dictated by several genetic and epigenetic marks, and several technologies like 3C methods, CRISPR-I, QTL studies, and more recently correlation studies have contributed to the knowledge that we have today. And we wanted to explore using a complementary approach that leverages long-read single molecule epigenetics to simultaneously map accessibility, methylation, genetic variants, and CTCF binding all on the same molecule of DNA. As we know, almost all epigenetic mapping studies have relied on short-read Illumina sequencing, and like ataxic or ChIP-seq, they all can detect regulatory elements, but they detect them in isolation. And we wanted to explore if with long reads, we could phase regulatory landscape and allow us to effectively distinguish alleles, evaluate the interrelationship, interdependence of elements, and also report on single molecule dynamics. And just a brief introduction to Oxford Nanopore technologies. With this sequencing platform, you can sequence native DNA or RNA and not only get the primary sequence, but also detect variants, detect modifications like 5-methyl-C, which is the one that I'm going to focus on today. You can phase haplotypes and assemble de novo assemble genomes and also specific loci of interest, large loci. Depending on how you plan your experiment, the cost is similar to what you would spend with Illumina sequencing, and you can sequence very long reads with an average of 100 kb, also depending how you prepare your sample. And one important point that I wanted to mention here is the accuracy, which has been historically in the low 90s, but in the last few years, it has increased to high 90s, and actually now the raw read accuracy is over 99%. And this is accessible for many labs with cheap cost when you do small scale, and also can spend more money as large scale experiments using the more expensive flow cells and devices. So our method works by first marking, not cutting, but we mark open chromatin in intact nuclei using a GPC methyltransferase. This enzyme methylates within nucleosomes and leaves a footprint for accessibility. And we then very carefully, isolate the high molecular weight DNA, and each single molecule in the sample now has both the native endogenous DNA methylation at CpG denucleotides, but also the exogenous GPC methylation at accessible sites of chromatin. And now, in order to sequence entire loci of interest at very high coverage, we use a pool of Cas9 guides flanking the regions to be sequenced. And we can then specifically ligate sequencing adapters to the molecules with new Cas9 cut ends. This is an example experiment in T cells where we excised 24 immune loci, and the loci were between 50 and 120 kb. Here in the middle, you can see that we get up to 30% of full length reads, and we can also get a very nice coverage, which for most of the loci is over 100x coverage, which is about 200-fold enrichment compared to a whole genome sequencing run without targeted enrichment. So then, together with Jingyi Wu, another postdoc in the lab, and Kevin Dong, a computational analyst at the Broad, we looked at this GPC methylation patterns, and basically to summarize, two things are important for us. One is this long stretches of GPC methylation that correspond to accessible regulatory models, but also this very short protected regions with no GPC methylation that we call short protected runs that are indicative of TF binding events. And those are different from the larger stretches of protected regions with no GPC, which corresponds to nucleosome. As you can see here at the length distribution of the unmethylated runs. Okay, so for the data then that we collected, we always looked for the short protected runs and also for the open runs that indicate accessibility. This is how the data looks like at single molecule level. This is a 72 kb locus in GM cells that contains this immune genes NOD2 and CYLD. These are over 100 single molecules The horizontal lines are molecules. In the first part, I'm highlighting in red the DNA methylation at CPGs. And then for the same molecules, you can see the accessibility. And then in the third part, I'm highlighting the short protected runs that show us the TF footprints. And we can, so you can see, for example, at the CYLD promoter that's unmethylated, accessible, and it has some TF footprints. And we can aggregate this data and create sort of pseudo bulk, and this nicely correlates with gold standard data. So here you see our methylation data compared to bisulfite, and the accessibility compared to DNA-seq. And then for the TF footprints, so here you can see two main footprints at CYLD promoter and shortly downstream. And when we did motif analysis and looked at ChIP-seq data, we saw that these footprints actually correspond to RFX5 and also CTCF further downstream. Okay, so we next examine the dynamics in chromatin accessibility of selected immune loci over the course of T-cell stimulation. This is here how we stimulated CD4 T cells using CD3 and CD28 antibodies, and we checked for the presence of those activation markers, CD25 and CD69. We collected data for these 3 time points, and we enriched for several immune loci in these 3 samples. And I'm going to focus on one of our favorite immune loci, which is this large locus that contains CD28, CTLA4, and ICOS. This is the part that we targeted that contains the CD28 gene. And when we looked at the accessibility, we saw this, especially at the 3' UTR, this highly dynamic part in accessibility. So we focus on the first part of this locus. We first annotated the putative regulatory elements, and we sort of reason that correlation between the pair of elements could reflect functional interactions. So we scan the loci to identify accessibility that correlates across the single molecule reads, and we identify the 7 pairs of sites that correlates across single molecules. Then we did PCA to cluster the single molecules on the basis on accessibility, and we can see that the 0 time point nicely separates from the 48-hour time point, and the 24-hour is right between the two. And then we apply T-scan, which is an approach that was originally developed to reconstruct single-cell transcriptomic trajectories. We apply this to single molecules, and we can see we detected these 2 states. And then what we did is to replot this each of these molecules, and we were able to reconstruct the time of activation. This is here. The order of these reads is based on the pseudo time, and this is the real time of activation, and those really nicely correlate. Okay, so but now I want to turn to some biology that's also a nice demonstration of the technology, and this relates to parent of origin imprinting, where a number of loci that contain several genes that are expressed either from the paternal or from the maternal allele. And so we collected data for yes cells and muscle cells, targeting 3 imprinted loci that are relevant for human diseases. Those are SNRPN genus and the IGF-2 H19 locus. That's the one that I am going to focus mostly today. So this is how the data looks like for this imprinted loci. This is a 116 kb locus that contain the SNRPN gene. SNRPN is expressed only from the paternal allele. So what you can see here first is the aggregated pseudobulk profiles for accessibility and DNA methylation. We are able to separate them into the two alleles using the heterozygous SNPs that we can very easily detect on this very long molecules. And basically, I'm here going back. So first, I'm showing you the profiles, but actually, if you look into this 3 zoom in parts here, you can see that they all contain this heterozygous SNPs that were used to separate the two cluster of reads. And basically, you can see that the paternal allele is highly accessible and the maternal allele is highly methylated, as expected for this particular locus. And this phasing here was done using the heterozygous SNPs. But what we also explored is using the methylation pattern itself and ignoring the SNPs to cluster them into different haplotypes. And this is shown here for the genus locus. Works also very nicely. It corresponds to the genetic phasing, also is shown here. So, but this is the locus that I really want to talk about, the textbook imprinted locus IGF2 age 19. So surprisingly, when we line our long read data to the AG38 assembly, we noticed that our reads did not align. There are two main errors in the AG38 assembly. One of them was the inversion of 2 genes upstream of age 19. There's 2 that are in the sense direction. They are actually one of them is annotated in the antisense, and the order is wrong. And we were able to correct this using our reads that we collected for several cell types and some tumor samples, and we confirmed this by looking at the new T2T assembly. And the second error was the segmental duplication that is actually is a 100 kb segmental duplication that covers the entire over half of the entire locus. And this was a big problem, but because when you look at all the ENCODE data, or actually really nice micro-C data, this segmental duplication is removed from the alignment, from mapping. And one of the things is here, you can see this artifact contacts because of the inversion of these 2 genes. And also you cannot see any contacts within this, the IGF2 locus. But if you remap the data to our repaired assembly, it's really nice, all these contacts that you can appreciate for this locus. So we corrected the assembly, and then we were able to carefully look at our data for this locus. But I just wanted to revise how this locus worked. So the difference between the 2 alleles, paternal and maternal, is not at the sequence level, is the imprinting control region that's different. So in the paternal allele, you have this CTCF sites that are methylated at the paternal allele. So this upstream enhancer can contact IGF2 and activate IGF2. However, the maternal allele, the ICR is unmethylated and bound by CTCF, so that the enhancer is blocked from contacting IGF2. So IGF2 is paternally expressed only. So here's our data. We collected data for ES cells and muscle myoblasts. And first, I'm showing here the DNA methylation for paternal and maternal alleles, and then the accessibility again for both cell types and both alleles. And we can see here when you look at the ICR, you can actually zoom in into the ICR and nicely appreciate that the paternal allele is highly methylated and inaccessible, and the maternal allele is hypomethylated and highly accessible. And especially here for the muscle data, you can appreciate that the maternal allele, the 7 distinct CTCF sites that are, this is one of the differences to the mouse model, for example, that only has 4 CTCF sites, and these extra CTCF sites are rows through the segmental, this duplicated sequence here that we also were able to detect. And another thing that we identify was this human-specific LPR that's highly accessible on both alleles. No, sorry, that we think, we speculate is here in order to shield this embryonic specific enhancer that we could see here on both alleles. Okay, so, but this was all fitting into the model. But now we ran into a little problem. We looked across across this region, and what we identified is this accessibility, this highly accessible region, especially in the muscle cells, that is much closer and proximal to IGF-2, and is on the same side than the ICR. So when we looked at bulk K27 acetyl data, we saw that there are enhancer-like features for the muscle cells and not for the embryonic stem cells. So we speculated, we hypothesized that this region here that's highly accessible might be a non-canonical element that could enforce biallelic expression of IGF-2, because we can see in our data that it's on both alleles, and there is K27 acetyl, and it would not be blocked by the ICR. And indeed, when we had the nanopore data, the phase nanopore data for this muscle cells, and within the last exon of IGF-2, there are 3, for this particular adult individual, there are 3 heterozygous SNPs, 2 on the maternal and 1 on the paternal. When we did RNA-seq of this data, we detected all 3 SNPs, which highly demonstrates that indeed IGF-2 is expressed from both maternal and paternal alleles. And this is different when you look at yes cells. This is a yes cell line with also an heterozygous SNPs. When you look at whole genome sequencing data that's available, and then RNA-seq, you only see one type of nucleotide of the RNA-seq. This is fitting the classical model that IGF-2 is only expressed by one of the 2 alleles. And we can see a similar thing in stimulated T cells. When you look at the K27 acetyl profile in CD8 resting T cells, there is no activity of this non-canonical enhancer, but after activation, you can see this peak signal of K27 acetyl. And when we look at RNA-seq data, we also can see that actually IGF-2 is expressed from both alleles and is not expressed on the resting T cells. And we see this enhancer, this novel element, in a number of cell types. These are the ones that I'm showing here. This is they have the enhancer, and we also confirm biallelic IGF-2 expression. This element is relevant for in human diseases. It's associated with several traits, human traits. And finally, we use CRISPR-I to validate the function of this enhancer. So if you look at the last knockdown here in HSMM and this fibroblasts, it has twice the effect. When we knock down the non-canonical enhancer, it has twice the effect in reduction of IGF-2 expression compared to the canonical enhancer, which really highly suggests that this non-canonical enhancer is active on both alleles. So back to the chromatin conformation. Here you can see this is the micro-C data realigned to our new assembly that I showed before. This is the schematic of the locus, and this is the novel non-canonical enhancer that we introduced. And we are, you can see that this is within the same pad as IGF-2, showing that it really can activate both alleles. You might ask, you might wonder why this element was not found in the classical mouse studies. And the answer is this non-canonical enhancer arose in primates at some point, and it's not in the mouse. As you can see here, is the mouse locus is much shorter, and it lacks the whole this whole region here. So just to conclude, we propose sort of a revised model of IGF-2 imprinting. This is here the classical model whereby the canonical enhancer is only able to contact IGF-2 on the paternal allele, because on the maternal allele, it's blocked by CTCF. So this is a paternal specific expression. But now we show that there is this other enhancer that's proximal to IGF-2 and is able to activate it on both paternal and maternal alleles in certain proliferative tissues and cell states. So to wrap up, I showed you how we can generate maps of DNA phase, DNA methylation, accessibility, and 3F footprints across these very long DNA molecules. I described how we can use this to understand the interrelationship among elements. And finally, how we can use this to understand how I showed you some specific features of this imprinted locus and a novel enhancer that drives biallelic IGF-2 expression. And I really like to thank the people involved in this project, members of the Bernstein lab, and especially Jingyi and Kevin, who led the computational analysis. And thank you for your attention, and I'll be happy to take any questions.
Amazing. Thank you for the wonderful presentations. Maybe we can have a virtual round of applause. So while we are waiting for questions to be typed in a chat, we have some questions. I would like to start with my question. I'm wondering if are there any possibility that the open run methylations, the methylation that you provoked in the DNA strand, can be confused with endogenous methylation? Is there an error rate for one type of methylation being read as the other one?
Yeah, that's a great question. So we looked for the nanopore can distinguish, like you read from 5' to 3', and then the CPG indicates the endogenous and the GPC the exogenous. But if you have a CGC, for example, then a GCG, you don't know if it's endogenous or exogenous, so we have to remove those. But those are about 10%, and so it doesn't really affect the the readout. And there is, the nanopore has an easier time detecting GPC than CPG methylation, but we take that into account.
Amazing. Yeah, 10% is not a lot. Okay. The second question is, it looks like you saw a biallelic expression of IGF-2 in many cell types. So is the imprinting effect only restricted to stem cells, embryonic stem cells? Do you have any idea of what could explain this pattern?
Yeah, no, it's not only restricted to the embryonic stem cells. So here, in this, here I'm showing pink one, which is those cancer cell lines are also, they also fit the classical model. We don't know really. We think that this proliferative cell types that need more of the growth factor are the ones that with this biallelic IGF2. We don't really know yet.
Okay. We have a question in the chat. Carl de Boer is asking, the resolution of footprinting is limited by the specificity of the methyltransferase, so the GPC. Are there other DNA modifications you could use that would increase your resolution?
Yeah. Hi, Carl. Yes, I guess. Well, one would be 6-methyl-A. I think that would be great. However, so there are some advantage and disadvantage of using Nanopore against other long-read platforms. We went for Oxford Nanopore, but one of the disadvantages that it has a hard time detecting methyl A from non-methyl A, so that's why we didn't use that one. But I think since it's advancing so rapidly, the field, I think soon we would be able to use other methylation marks that would increase the resolution for footprints. Yeah.
Okay, thank you so much for the questions. I think we don't have any more questions in the chat or the Q&A, so maybe we can go ahead with the second speaker. Thank you, Sofia.